Dear Professor Roska, dear Mr. Rector, Prorectors, and dear our guests. Today is dedicated to the vision and to the work of Professor Roska, and I would like to tell about the clinical aspects, what he is experimenting, about the inherited retinal dystrophies. And in the next part of my talk, I will talk about precisely about the retinitis pigmentosa, which is in the focus our experiment. In the background, the definition of the retinal dystrophies, they are a group of genetic diseases with primary involvement of the photoreceptor cells, which you saw in the previous presentation, with progressive nature and high degree of heterogeneity. The prevalence is quite high, 1 to 3,500 to 4,000 worldwide. In the Hungarian patients, the estimated number, about 2,500, 3,000 adults are affected in the adult onset form. The most severe cause symptoms before the first year of age, this is the Leber congenital amaurosis, so the most severe form of retinal dystrophies. And the number of Hungarian Leber congenital amaurosis patients, the number estimated between 200 and 250. So it's not very high, but it's a very bad disease. The number of identified causative genes is constantly growing. So here you see that this is what we identified during the past years. And retinal dystrophies are characteristically monogenic diseases, so affects usually one gene. The pathomechanism may involve at least 3 different pathways. The first of all is the photoreceptor structure. The second, the phototransduction system, and the retinoid cycle. The inheritance pattern, it is usually sporadic. In 50–60% of the cases, no affected family member. The other is autosomal dominant. It's about takes 25%. The affected patients in 2 consecutive generations in a family, both sexes may be involved, so men and female as well. The third one is autosomal recessive. No known affected family member in the previous generation, more common in consanguineous families. And the fourth form is X chromosome linked in 10 to 15%. Female carrier, male offspring is usually affected. The affected photoreceptors, the rods, it is the problem is in dark adaptation, so in dim light vision they don't see at all. The cones, they are in sharp and color vision, so the daylight vision is usually affected. In the foveola centralis, you also saw in the previous presentation, there are only cones. Rod density increases toward the peripheral retina. So here you see the rods represented by the green, and the cones represented on the left figures by the blue. So in the eye, rods are usually 130 million, and cones only 6 to 7 million. Cones are the most important for sharp vision. About the symptoms, about 90% of the retinal dystrophies show primary rod dysfunction, so which is in much higher in number. This is called rod cone type dystrophy, retinitis pigmentosa. The symptoms: night blindness, preserved central visual acuity for long run, but at the end stage this causes blindness, photoaversion in advanced stage, and concentric visual field narrowing. Part 2 to the symptoms. About 10% of the retinal dystrophies show primary cone dysfunction, so it's cone rod type disease. The symptoms: photoaversion, decreased central visual acuity, so they cannot read. They see with the periphery, but the sharp vision is very badly affected. Also with color vision impairment and the central scotoma. So this is the contrary what I've shown you in the previous slide. There are also early onset retinal dystrophies. In the most severe forms is the Leber congenital amaurosis. The onset is usually before the age year 1. Here you see three siblings. One is affected with the label congenital amaurosis, lack of visual behavior, she. And the second sign is the oculodigital sign, so they press their eye because then they get some visual, due to the pressure effect, some visual sensation. The other early onset retinal dystrophies, it is a type 2, onset after 1 year of age, slightly milder symptoms and distinct genetic background. This is the histopathology, rapid retinal remodeling and thinning with the disappearance of the outer retinal photoreceptor layer. Here you see the normal healthy retina, and then this is degrading in 6 weeks of time. This is the article which was quoted. Clinical examinations, routine eye examination, so the fundus photo documentation, and electroretinography. This is the most important, full field ERG. We don't have this device because it went wrong, and it takes a while when we can have another one, and this is very important in the diagnosing of this patient. This is the normal electroretinogram. And the second one is a completely extinguished, so the line is parallel, and there are residual responses, so this is the early onset disease. Visual field examination. So this is a normal field, and this is only the central field can be shown, concentric narrowing in retinitis pigmentosa in this patient. And finally, between the age of 50 and 60, characteristically, this is also lost, and the patient got completely blind. Retinal imaging is a very important new method. It's called optical coherence tomography. And this here we can see some structural changes in the retina. A foveal thinning and irregular residual retinal pigment epithelium, plus the photoreceptor layer also. Here you see in retinitis pigmentosa a severe remodeling in the outer nuclear layer, and also in the neuroretina shows cystoid intraretinal. Here this is the cystoid intraretinal spaces. Theoretical treatment options, not very much. Pharmacotherapy, only limited cases. Now it's mainly in dogs with the RPE65, 9-cis retinoid substitution, gene replacement therapy if retinal and macular structure is preserved. Stem cell therapy if photoreceptor cells are aplastic or irreversibly damaged. There are some human clinical trials after many years of experimental treatment in different animal models, zebrafish, mouse, and dogs. And active retinal gene replacement phase 1 or 2 trials are targeting the Stargardt disease, retinitis pigmentosa, X-linked retinoschisis, chorioderemia, achromatopsia, and LCA type 2, so the retinal pigment epithelial mutations. Professor Roska, as you heard, that he graduated in the Semmelweis University, then had postgraduate training in University of California at Berkeley University, Harvard Medical University, then Basel, Friedrich Michel University from 2005. He became a molecular and cell biologist and now a leader of the medical research group in neurobiology and for molecular and clinical research for vision. The 3 most important awards, except what he will get today, a bit later. Hopefully he will be so proud of that as the other 3, Bressler Prize, Alden Spencer Prize, and Louise Jante Prize. A research field, how human brain processes imaging information, understanding the mechanism of human vision, animal models for inherited and retinal dystrophies, new methodologies, in vitro human retinal cell cultures in cooperation with the Semmelweis University and with Dr. Arnold Szabo from the Department of Anatomy, synthetic viral vector for delivering gene replacement therapy. I think this should have been written in bold. So this is the most important at the moment, and also optogenetic therapy. About retinitis pigmentosa, which is now a field of interest. It's a group of inherited retinal disease with X-linked autosomal recessive and autosomal dominant inheritance. The most important part is the impairment of photoreceptor and the pigment epithelium. Symptoms: night blindness, progressive loss of peripheral vision, pigmentary retinopathy, abnormal electroretinography, and central vision usually affected later. This is one of the leading causes of inherited blindness, with approximately 1 to 4,000 individuals. Worldwide, 1.5 million people affected, so it's a big number globally. Early onset retinitis pigmentosa occurs within the first few years of life and is typically associated with syndromic diseases forms, while late onset retinitis pigmentosa emerges from early to mid-adulthood. This is the so-called diffuse bone spicule pigment clumping. Here you see it starts around the periphery, then it gets closer and closer, and on this picture you can also observe pigmentary changes, retinal arterial attenuation along the vessels, waxy pallor of the optic disc, so this is usually not so light, not so whitish, macular pigmentary changes. Here you see posterior subcapsular cataract, vitreal cells and debris, and cystoid macular edema up to 20 to 30% of the cases, which even more deteriorates the visual acuity, and abnormal electroretinogram and electrooculography. So far, about 150 mutations have been described, so it's not so easy disease to solve and to cure. Opsin gene may be affected with intradiscal, transmembrane, and cytoplasmic domain mutations. One of the main biochemical causes of retinitis pigmentosa in the case of rhodopsin mutations is protein misfolding and the disruption of the molecular chaperones. There are codon mutations, I don't go into details, and in the autosomal recessive inheritance patterns, 45 genes have been identified so far. About the pathophysiology, very shortly, mutations in the rhodopsin gene and problems in the phototransduction cascade. And the other pathophysiological problem, misfolding of the class II rhodopsin gene with two proteins, the transducin and opsin proteins, are damaged. In animal models, it is suggested that the retinal pigment epithelium fails to phagocytose the outer rod segment discs that have been shed, leading to an accumulation of outer rod segment debris. The diagnosis, progressive loss of photoreceptor cell function, visual field and visual acuity testing, and the most important in the early diagnosis, the ERG, the electroretinography. In the periphery, the so-called bone spicule accumulation of pigment clumps, and also with optical coherence tomography provides clues into photoreceptor thickness. With this method, we can measure with a micrometer precision the thickness of the part of the retina. In the retinitis pigmentosa phenotype shows decreased or delayed electrical response in the rod photoreceptors, as well as possibly comprised cone photoreceptor cell response. So the ERJ, I would like to suggest it's a very important examination method. Genetic counseling, it's very important that what to counsel for the patients who already have such kind of child. It depends on an accurate diagnosis, determination of the mode of inheritance in each family, and results of molecular genetic testing. There is no really cure at the moment for retinitis pigmentosa. The efficiency of various supplements such as vitamin A, lutein, and delaying disease progression remains an unresolved yet prospective treatment option. Clinical trials investigating optic prosthetic devices, gene therapy mechanism, and retinal sheath transplantation are active areas of the study. The father of Professor Roska experimented on the prosthetic devices, which was a very important target of the European Union. The goal of gene therapy studies is to virally supplement retinal cells expressing mutant genes associated with the retinitis pigmentosa. I think you will hear about in the next presentation by Professor Roska. And in the literature, I think we can say that response to gene therapy would theoretically benefit young patients who still have a good function of the optic nerve and exhibiting the shortest progression of photoreceptor decline, thus correlating to a higher possibility of cell rescue via the healthy inserted gene, which should go into the vitreous part of the eye through an injection, which we already administer with the anti-VEGF therapy for patients with macular degeneration. Future treatments may involve, so as I told, retinal transplant, artificial retinal implants, gene therapy, stem cells, nutritional supplements, and or drug therapies. Thank you very much for your attention.